You know, for as long as we've been human, we have been at war with a single germ. It's a conflict that started long before any empire we can name, and it's a battle fought not on fields, but deep inside our own cells. This is the story of tuberculosis, or TB, humanity's oldest microbial enemy, and the incredible new science that might finally let us win. I mean, just listen to that name, the captain of all these men of death. That's what they called TB centuries ago, and it really captures the sheer terror it inspired. Before we even knew it was a germ, it was the White Plague, named for the ghostly pale look it gave its victims. Or it was called consumption, because it literally looked like it was eating people from the inside out. At its worst, it was killing 1 out of every 4 people in Western Europe. And this isn't some dusty history lesson. That number you're looking at right now, 1.25 million, that's how many people died from TB just this year. To put that in perspective, that's more than HIV and malaria combined. The captain of death is, believe it or not, still on the march. So how did this ancient plague become such a modern crisis? Well, to really get a grip on the challenge we're facing today, you have to appreciate the enormous timeline of our relationship with this bacterium. Okay, the scale here is just wild. 3 million years ago, the ancestors of TB were already infecting our early hominid relatives. For basically all of human history, we had no idea what caused this disease. Then, in 1882, Robert Koch finally puts the culprit under a microscope. And by 1943, we get our first antibiotic, streptomycin. And we really thought, okay, that's it, war's over, we won. But as we all know, the story did not end there. So what makes this germ so darn resilient? Well, the answer is that it's a master manipulator. It plays our own body's defenses against us. And the main stage for this whole drama is a tiny structure in our lungs called the granuloma. So when TB gets into the lungs, our immune cells do what they're supposed to do. They rush to the scene and they form this organized little ball, the granuloma. You have the infected cells in the middle and then a wall of other immune cells surrounding them. For a very long time, we fundamentally misunderstood what was actually going on inside. And this is probably the most important shift in our thinking. We used to see the granuloma as a fortress, our body successfully building a jail and locking the invader up. But we now know it's not a jail at all. It's more like a niche, a safe house that the bacteria actually helps build for itself. It's a place to hide from the immune system, go dormant for years, and, this is the crazy part, lure in fresh, healthy cells to infect. And here's exactly how it pulls off this heist. So an immune cell called a macrophage, its whole job is to eat and destroy invaders. It swallows the TB bacterium. But once it's inside, TB works its magic. It literally stops the cell's digestive system from kicking in. It's like it disarms the bomb. So instead of getting destroyed, the bacterium is safe, it multiplies, and eventually it can even break out to infect more cells. It turns our own bodyguards into its personal incubators. But here's the good news. The science has not been standing still. In just the last decade, our ability to fight back has been completely revolutionized, from the way we diagnose TB to the way we treat it. Just check out the difference in diagnosis. It used to take weeks, sometimes months, for a lab to grow the bacteria just to confirm someone had TB. It was agonizingly slow. Today, we have modern molecular tests like Expert Ultra. They can spot the bacteria's DNA right from a patient's sample in less than 2 hours. That's not just an improvement. It's like going from using a compass to using GPS. And the change in treatment is even more incredible. Patients used to face years of isolation in sanatoriums, then get hit with these complex, toxic drug cocktails with painful injections for 18 months or more. Now, we have all oral regimens, just pills that can cure even the most drug-resistant forms of TB in only 6 months. This is hands down one of the greatest public health achievements of our time. But there's a catch. Because just as our weapons get better, the enemy evolves. The single biggest threat we face today is the rise of drug-resistant TB. Now, a lot of other superbugs can actually trade resistance genes with each other, kind of like trading cards. It helps them get stronger faster. TB does it the old-fashioned way. It relies on random, spontaneous mistakes, typos, in its own DNA. So if a course of treatment isn't perfect, the few bacteria that happen to have a lucky genetic typo survive, and they're the ones that multiply, creating a whole new army of resistant germs. Here are 2 perfect examples.
Our best drug, isoniazid, has to be activated inside the bacteria by an enzyme called Kg. But a single typo in the gene that makes Kg means the drug never gets switched on. It's useless. For our other workhorse drug, rifampicin, a tiny mutation in its target changes the shape just enough that the drug can't grab on anymore. It's like changing the locks. It's so simple, so elegant, and for us, so deadly.
And the scale of this problem is huge. This year, there are an estimated 390,000 new cases of TB that are resistant to our most important drugs. Every single one of those cases is harder to treat, way more expensive to treat, and far more dangerous. So you might be thinking, this sounds a bit grim. We're facing this ancient, super adaptable foe. But here's the thing. For the first time in a century, there is a powerful new sense of hope. And it's being driven by a whole new generation of tools.
And that really is the question that defines this whole era in the fight against TB. After millennia of being on the defensive, can we actually go on the attack and, eliminate this disease for good?
The biggest missing piece in our arsenal has always been a good vaccine. The one we have, BCG, is over 100 years old. It provides some protection for young children, but it does almost nothing to stop adults from getting infected in their lungs, which is how the disease spreads. But look at this. This chart shows the breakthrough we've all been waiting for. New candidates, like one called M72/AS01E, have shown around 50% efficacy in clinical trials at preventing adults from getting sick. 50%. That is a total game changer. A vaccine that could cut the number of new cases in half could finally break the cycle of transmission.
And this really brings it all together. For the first time in history, we have a complete toolkit. We have new vaccines to prevent infection in the first place. We have new diagnostics to find it and figure out its weaknesses in hours, not months. We have these amazing new 6-month all-oral cures. And we have a pipeline of new drugs that attack the bacteria in totally new ways. This isn't just an upgrade, it's a complete paradigm shift. So, the battle is no longer purely scientific. We have the tools. The unkillable germ is for the first time truly vulnerable. The final challenge isn't about discovery anymore. It's about delivery. It's a question of will, of investment, of equity, of making sure this revolutionary science reaches every single person who needs it. The war isn't over, but now, for the first time, we actually have a clear path to victory.